And our first speaker is Joachim Schultze. Joachim just recently became a director of systems medicine at the Helmut Center DZNE. And in parallel, he holds a position as a department director for genomics and immune regulation at the Limes Institute. And last but not least, he is our speaker of the Central Coordination Unit of the NGS Competence Network. So, and he is going to introduce you into his last initiative that he started together with the other NGS competence centers in spring as a answer to the challenge of the COVID-19 pandemic. So Joachim, I would like to ask you to share your screen.
1 second.
Actually, we are a little bit ahead of time, but I think we will lose time over this session. So that's why it might be.
There's also one housekeeping rule I forgot to mention. So all the participants should ask their questions in the Q&A session, in the Q&A section of the session. We will then ask the speaker those questions after the talk. Sorry.
So Peter, can you see my screen? Is that, yeah?
Good. I think it's 3 minutes ahead, but are we afraid that some people would just come in when you start?
No, it's fine. It's absolutely
Otherwise,
fine.
I think we should start.
Yeah, so Peter, thanks for giving me the opportunity to speak this year and to have this unfortunately timely content and subject. So I overrode this with DeCOI meets the Arbeitsgemeinschaft. So DeCOI stands for Deutsche COVID-19 OMICS Initiative or German COVID-19 OMICS Initiative. And this is really an initiative, meaning we are a bottom-up approach where people came together thinking that we can use our expertise and our knowledge to really help to find solutions to the crisis. And this all started with some thinking in February and March, really saying like genomics can help to solve the pandemic, and the wish to say we need to do something. And this was in February and March, and there were some discussions ongoing. It started slowly, but then in 22nd of March, I decided as the speaker, as Peter alluded to, as the speaker of the NGS Competence Network, just to call my colleagues and say, like, have you already started to think about using genomics? This is like in Kiel, Philipp, Rosenstiel, Ezio in Dresden, Olaf in Tübingen, Peter Nürnberg in Cologne, Dagmar Wittrek in Düsseldorf, and Markus here in Bonn. And Miriam is our organizer, and asking, would you like to collect information? And I also asked them on a Sunday morning, would you have time the next day in the afternoon to speak? And I have to say this was probably one of the most intense weeks this year, because just a week later, Monday, March 30th, we had basically written up 8 projects. We got some DFG support to basically start the centers to do experiments and to do these projects. And I think that was an enormously tense week. We decided that there are 3 areas that we would like to work on. And then from there on, it started. And we had discussions with others in the field. Very quickly with the people and our colleagues from the German Human Genome Phenome Archive Consortium, which was at that time not funded. It was a consortium in space, so to say. And then it all came together that we said, like, we need to basically join forces and then come and work together. And I have to say, it's a really nice thing to see at the end of the year that we now have 88 members. As I said, there's no funding directly to the network. It's really a group of scientists and citizens who bring their knowledge and expertise to the table and work together on questions concerning COVID and the SARS-CoV-2 virus. And I'm very proud to be part of this because it tells us that science can be done in a completely new fashion, and in my view, much more collegial than what I have ever experienced before. And I'm actually a friend of teamwork, but this is much more than I ever have seen before. So who's coming together in DeCOI? Many experts that can do everything from the sample to basically the analysis of omics data. So we have a lot of people that know the upfront and can help the clinicians and the biobankers to know how to deal with these things. We have sequencing in the network. We have more than 10 Novaseq, so it's really powerful network. We can, cross the board from genomes, transcriptomes, epigenomes, single cell up to HLA. That was all part of that. Data management, automation, pre-processing, workflows, extended data analytics, and then once GHG8 got them to know that it's going to be funded, we have even a very strong network partner that can leverage all the knowledge that has been gathered in this network as well. So in the beginning, we said we want to have a couple of structural as well as strategic and scientific goals. And it's very interesting if you now look back 9 months later, many of the things we have actually achieved. We had wanted to develop a network of all major academic centers focusing on sequencing. That has been achieved. Connect to the GHG8. This is really integrative, very nicely happened. Provide multi-omics capacities for all national and international activities that we can see. We can do that, and we have done that already. It could be more. Contribute sequencing to the first wave of the pandemics, that we have certainly done in Germany. Prepare networking for second wave. We are now in negotiations also with additional networks that we can provide our expertise. Build long-term infrastructures. Also here, the NGS Competence Network just learned, and it's public on the DFGNet website, that we have a continuation of 2 years, and I'm pretty sure our efforts with DeCOI have helped to support that decision also by the DFG. And then there is, of course, a new culture that we would like to have, because many of you in the audience, I know, they must have laughed after a while when I opened up every meeting with the major concept is sharing, sharing, sharing. And in the end, this is what happened and what made us strong. We have 3 major scientific goals. One was around the viral genome and also metagenomes to learn more about the virus. The other one was about the immune response, to take functional genomics, to learn more about the immune response in COVID-19, and then a large group that focused on host genetics. So we had a clear picture, and in these 3 areas, we had pushed out 8 projects, and then there were more that came, the larger DeCOI was growing. Now, why is this all important? And I just give you snapshots, because the whole afternoon you will see fantastic experiments and exciting projects that came from this. But I give you a little bit an overview why we think this is so important. Viral sequencing is super important. First of all, if we would not have sequenced the virus from the beginning, many of the aspects would not have been known at all. Even if you think about the vaccines on RNA basis, you need the sequence of the virus. If you don't, otherwise you cannot even design the vaccine. So I haven't even talked about that. I just give you here the major things for the future probably is that with sequencing, targeted outbreak investigations is so much more powerful than if you do just a PCR test and have a positive or negative, because here you can really follow who infected whom, at which time, at which location. That could also all be done. And there are other countries out there that are much further ahead, but we're working ourselves into this, and we will hear a little bit about that today as well. Systemic outbreak investigation could be done, and we would be ready in essence. Yeah. Then monitoring the vaccine resistance. Some virologists say that's not so important. I would be more open, if there is evolutionary pressure by a vaccine on the virus, there could still be that we might see this. And if you do viral sequencing, you could find that out quickly. And of course, the monitoring by PCR testing, if the test is not positive, that could also be because there's a sequence that is changed in the virus and then not detected anymore by the simple PCR test. Why is human genome sequencing so important? Well, we learned during the summer, and we will learn more here. So I just took the other side of the coin. You will hear more from Andre Franke about what they found. But there's also indication that many of the important pathways that we know, like the interferon pathways, seem to be changed and mutated in some of the patients, and these might be then more severe patients. And functional genomics is super important because we need to know what kind of pathways are switched on. And this is just a snapshot out of one of our single cell papers that came from DeCOI, where we can clearly show that if you take a patient over time, these are single cell data, violin plots, you see that you always lose the signal of interference signatures is always going down in every single patient, and that's true for mild as well as severe over time. At that time, when we learned this in summer, nobody knew about this. And so this was very important because there was some issues about immunological testing that had been done before, and we, with using functional genomics and single cell, could clarify that. So this is an example. So DeCOI with the NGSCN has a lot of things on the plate. Viral sequencing, about capacity building there, immune response. As I told you, there's also many of us are still working on TCR sequencing and on single cell. We have published, we will hear about this, multi-omics clinical trials, and the whole work on host genetics is ongoing. We connected, this is also important, and it was a very nice experience as well with many others in the world. GHD I mentioned, the German biobanks, also international consortia, GIS8 and NextStrain. So many of the colleagues had their connections and used them to connect DeCOI to the world outside of Germany, which I think is very important. Now, it has been fruitful, and this is, of course, only a small list of reports from DeCOI members, and this is growing, and I'm pretty sure that we will continue to contribute, really illustrating how important genomic technologies are now to really identify new diseases, characterize them, and also the virus that causes it. Now, I would just give you a little snapshot in the last 5 minutes that DeCOI goes actually beyond. And that was an experience that I had just in the second half of the year. We had the chance actually here in Bonn to work together with Hewlett-Packard Enterprise and Dutch and Greek colleagues to work on something what is called swarm learning, and I will introduce that in a second. And we had basically a small set of samples from Athens and from Nijmegen to find out a very, a very straightforward but then also difficult question. Could be basically in knowing that there's a PCR test, but could we detect patients' disease? And not only that they're infected, but can we detect patients' disease, and could be used for that transcriptomics, blood transcriptomics. And on top of that, could we build classifiers by machine learning, and could we do that quickly without even exchanging data? That was the questions, and everybody would say like, well, seems to be a little bit too many things at once. And so the concept of swarm learning is then basically, the technical basis to that is that if you take the scenario here in the middle that you have, let's say, a hospital number 1 and a hospital in another city number 2 and number 3, that they might not be able to exchange the data because of data protection laws and so on. So could we make a system that one hospital learns on their data, a second hospital learns on the other data, and a third one on their data, identifying, for example, COVID and controls, but could we then bring this all together without sharing data, only sharing parameters? Yeah, and that's the concept of swarm learning. And this is what we try to do. And since we had only 2 sites, we split up the Nijmegen data more or less into 2 sites and the Athens in another, and then asked whether this is working. Yeah, so the setup was basically this. At the beginning in May 2020, a couple of patients from Nijmegen and Athens. And then we did this, and without showing the data, just believe me, we got a fantastic classifier for COVID-19 just on blood transcriptomics, really identifying these patients. And then in July or end of June, we sent out the paper, and knowing that we will get back questions, and it came back and took enormously long. On the 14th of September, like 10 weeks after we had submitted this paper, the editor apologized, so it took too long, but has been now seen by the referees, who comments are attached below. While they find your work of potential interest, that's what we expect. as do we. That's nice. They have raised important concerns that in our view need to be addressed before we can consider publication, as we are known to these kind of answers. But the overall was a very positive response. Now, if you have to do extensive additional work, this can only be done if you have a network. And here DeCOI comes in. And that was a fantastic experience because, suddenly when we prepared our colleagues and said, like, we're expecting that there will be these questions. Could you help us if they ask us for doing more? And basically the whole DeCOI, whoever had samples from Kiel, from Cologne, from Aachen, Bonn, Saarbrücken, Tübingen, Munich, all basically said, yes, we're joining in. And now we're going to do this all over and make an even more realistic scenario and show that we can do machine learning-based, transcriptomics-based classification of this disease. And if we have enough samples later on, we can even think about, can we identify very quickly the severe ones by this technology. And so this is what we did. And for some of you who have also given us samples, this is probably the first time you see the data. We sent around the draft that's going to be submitted hopefully before Christmas. But here's the principal idea. So now every node here is a city. We didn't put in the names, but you can see that there's Kiel, that there's Tübingen, Munich, there's Saarbrücken, and so on. And then we basically asked, what happens if you learn only in Kiel or only in Tübingen or only in Munich, or if we learn together as a swarm, and how are the results then? And you can see here then in this graph is the different statistical tests for such a classifier, the AUC, the accuracy, sensitivity, specificity, F1 score. And in principle, what you can see is that, if you learn alone and then you get new data that the place has never seen, you can see that learning alone sometimes does not lead to very good results. Yeah, and here there are examples, for some where this is really not going well because, yes, it's very hard to learn on very few samples. But just think about, here we simulated it, but in essence, the system is now set up that we could leave the data at your place and then build this swarm. And you can see the swarm has enormously good statistics. It outperforms. If you look at overall, it outperforms every node. Because the combination is important, and the most important one is certainly the AUC. And you can see that it's really what makes the difference. So DeCOI, this is the power of DeCOI. Yeah? This would not have been possible. And that's why I thank all the members of DeCOI very much. There's so many, I cannot say who else, I just say for everybody in DeCOI, thank you for this year. I think we can continue next year, and I hope to see you all then in our meetings and talk to you. Thanks for this year of this journey. It was an amazing journey, and only as a team we were really successful. Thank you.
Thank you very much. It was a great talk, and it was really impressive to see how fast this important network developed. So, and how successful it already has been. So the question is, what will be the lifetime of this network if it is depending on SARS-CoV-2?
Yeah, so I think DeCOI is an example of how we can bring expertise together. And of course, we will change the name, but my wish for 2021 and the years to come is that we demonstrated how genomics could be done in Germany, and that's what we're working towards. Yeah, so leveraging the power of the many rather than fighting each other, and I think that would be a concept. So DeCOI, once the pandemic is gone and once SARS-CoV is gone, we might stay a little bit longer just to finish our papers, but then it should be changed into how the genomic community should work together in Germany.
And what about the long-term effects of the disease?
That's a good point. So there are several of the members in DeCOI, together with clinicians and clinical partners, are now thinking about studying that as well. And there have been already projects aligned. So I'm pretty confident that genomics will also contribute to that in many different ways.
Okay, thank you again. Very good initiative.